The materials used were chromatography paper, spinach, and solvent. In step 1, a line was drawn 2 centimeters from the edge of the chromatography paper. Throughout step 2, spinach was placed on each X on the line. After the spinach was on the line, a quarter was used to extract the pigments from the spinach. In steps 3 and 4, the chromatography paper was rolled into a cylinder. Once the chromatography paper was rolled into a cylinder and the solvent was entered in the jar, the chromatography paper was put inside of the jar. Once inside of the jar, the chromatography paper absorbed the solvent, which then extracted the pigments from the initial spot on each X. Throughout this next process, you will see the solvent traveling up the chromatography paper. Once the solvent reaches the top line on the chromatography paper, the paper is then removed. Once the chromatography paper is removed, the colors of the pigments can be analyzed. Each color represents a different pigment. In step 6, the distance of each pigment is measured and recorded. This lab showed that the pigments traveled up the chromatography paper, resulting in colors of orange-yellow, blue-green to bright green, and yellow-green to olive green. Performing this lab allowed the separation of pigments from the spinach. These results allowed an analysis to be performed by calculating the distance pigment migrated over the distance the solvent migrated. Through this lab, it was determined that the beta-carotene pigment was yellow-orange, the xanthophyll pigment was orange, and the chlorophyll pigment was primarily green with a tint of blue.